Welcome everybody. Thank you so much for being here. My name is Jill Metlin, and along with Sandy Shapiro, I'm the co-chair of the Society of Healers. This is my third or fourth year. They can't seem to get rid of me, so I just keep showing up. The Society of Healers brings us all together around our own way of providing tikkun olam in the medical community, and Federation in general is here for the entire community to strengthen, serve, and support, and help you feel more connected to our local Jewish community and to the larger Jewish world. By joining us this evening, you'll hear the most current information on COVID-19 and the vaccine, and we look forward to learning more from Dr. Michael J. Koren. I'm going to do a brief introduction of Dr. Koren, and I say brief because his accomplishments are very long and prestigious. He is a practicing cardiologist and the CEO for the Jacksonville Center for Clinical Research. JCCR conducts clinical trials at 8 locations in Florida. He received his medical degree with honors from Harvard and completed both his residency in internal medicine and fellowship in cardiology at New York Hospital Memorial Sloan Kettering Cancer Center, Cornell Medical Center. He's a fellow of the American College of Cardiology, past president of the Academy of Physicians in Clinical Research, and works with the American Heart Association. Dr. Koren has served as an investigator in more than 2,000 trials. He's been the international lead investigator for many multi-center trials with Alliance, Mendel, Treat to Target, Osler, and Roll. He has written and co-authored over 100 peer-reviewed articles. In addition, Dr. Koren has designed a research training course for physicians, now in its 20th year. And without further ado, Dr. Koren.
Well, thank you very much for the introduction. I appreciate it. And I'm really looking forward to tonight for a number of reasons. One is that this is one of our first lectures for a very long time where we actually have a live audience, which is really tremendous. And we'll talk a little bit about that in a moment. Let's get to the first slide, and I'm gonna lead off with a few introductory ideas here. And hopefully we're gonna have a really engaging audience interaction. We're gonna have a lot of fun, and we're gonna learn something about the COVID-19 vaccines. So the first question you may ask, and it's a very reasonable question, is what the heck is a cardiologist doing studying COVID-19? And so that's a particularly good question. And the answer to that question actually has to do with where we're standing right now. So we're here on University Boulevard, right near Memorial Hospital. And this is the first site for Jacksonville Center for Clinical Research, which is a clinical trials organization that involves over 100 physicians throughout the state of Florida that is operating clinical trials in 8 locations. And because of my expertise in running clinical trials, I had the good fortune of being asked by both Pfizer and Moderna to be the principal investigator for research on the COVID-19 vaccine here in Jacksonville. And I also am a sub-investigator for the Novavax study. And we have many other people in the room that have been part of all those trials. So they are very knowledgeable people, and they'll be part of this conversation today. So that's part of the reason why a cardiologist is talking about viruses. Obviously, viruses are something that we learn about in cardiology and internal medicine. It was mentioned that I did my internal medicine training at New York Hospital, Cornell. That happened to be during the HIV crisis. And during that time, I actually served as a chief resident, which is the same position that Dr. Anthony Fauci served at New York Hospital 20 years before I did. So I can say I had the same job as Tony Fauci. Although we went in different directions after that, and he's a lot smarter than I am. So here are the rules of engagement for tonight. So the rules of the room is that you see some people here, and everybody in this room has received the vaccine. So we are actually enjoying the dividends of having been fully vaccinated. And it's wonderful, and I feel very comfortable here in this room without a mask with all these people who've been vaccinated. But I will say that despite the fact that we're all vaccinated, we are still taking precautions. So you can see that the tables are set apart. None of us are interacting within 6 feet of one another. And we're being very respectful. We're not sharing drinks, and we're not kissing each other. So these are very important things in terms of social distancing. So although our risk of infection is really very, very low, and I'll show you the data, it's not zero. And there's still some risk of transmission that we'll talk about. But again, we're talking about very, very low levels of transmission risk. The other part that I'd like to let people know about is that we tend to teach in this organization using what we call the modified Socratic method. And for those of you that are not familiar with that term, the Socratic method involves asking questions. And you ask questions in order to elicit responses from the audience, and through that, learning occurs. So it's funny that the Socratic method was probably popularized in some things that you may have seen in the movie. If you remember the Paper Chase, the movie and the TV show from years ago, where John Huston, as the arrogant and intimidating law professor at a place that seemed like Harvard, was asking the people in the room who were prospective law students to become, to ask questions about things, and was incredibly intimidating. And this is our idea about the Socratic method, and it works to help people respond to difficult situations and to think on their feet. But it also is a really effective way for group learning, because not only do you learn when you're being challenged, but when you're being challenged, you seem to learn better about how you would think about something. So the natural human instinct is to put yourself in that place, and how would I answer that question? And then obviously the professor will go around and hopefully give you some background so you all learn. So I have found over the years that the modified Socratic method, where maybe not as intimidating as a paper chase, is actually a good way to share medical information and to learn collectively. So hopefully you'll agree with that, and this is a little bit of an experiment to see if that's true. All right, we can go to the next slide.
You got to break it.
Here we go. So we're all familiar with the fact that you had this horrible pandemic, and certainly the United States has probably been the worst hit country in terms of documented cases. This is the website that I like to go to on a day-to-day basis in order to get updates about the incidence and prevalence of this horrible infection, and also looking particularly at the death rates. You can see that this is a listing of all the different countries of the world, with the United States leading the pack with for this particular day, which I guess was either today or yesterday, there was 56,000 new cases that were reported, and the total number of deaths in the United States is about 530,000 deaths. So that's a horrible pandemic. But to put it into context, it wasn't quite as bad as the pandemic, for example, in 1918, with the Spanish flu, where there was over 1 million deaths at a point in time when the population of the country was only 1/3 of what it is today. So again, this is horrible, but when people say it's unprecedented, that's not true. We've had horrible pandemics before. So that, just a little bit of context there. And then the other thing that's interesting about this particular slide is to see the variability in different parts of the world. So some of these countries don't have the same kind of testing capabilities that we have in the United States. So if you look at the number of tests done in different parts of the world, you can see that in the United States at this point, we've done more tests than people. So there's been over 1 million tests per 1 million population. The only other country that's at that level is the UK, where they've done 1.3 million tests per 1 million people. And there's other places that don't have the resources that we have. So for example, in Brazil, which is a very, very hard hit country, they've only done about 1/10 of the testing of the United States. That obviously influences our knowledge about different places, particularly when we look at the impact of disease. So I'm not sure if our slide progression mechanism. All right, there we go. And then when you get down to the state level, I find this website to be really, really helpful. It's called Worldometer website. And this gives us the opportunity to compare our state of Florida with other parts of the country. And we actually stack up pretty well, really, really well. We'll get more into some of the details. But what I found really, really interesting is if you look at the most populated states in the United States, California, Texas, Florida, New York, you can see that the total cases are about the same per 1 million population, really remarkably the same, all about 90,000 cases per 1 million population. So across the board, about 9% of people in the United States have been infected with COVID-19. Now, keep in mind, these are only the documented cases. So there are many, many people who have gotten the virus but have never had the testing done. And the best estimates are that at least that number of people fall into that category, and maybe even more than that. And this becomes very important when we start talking about herd immunity. The other thing that's very, very different is that although the case rates are remarkably the same, the deaths per million population are actually fairly different. And we're going to go back and talk about that a little bit more, but you can just take a quick look at yourself and see that there's up to a twofold difference from state to state in terms of number of deaths. And certain epidemiological strategies may be responsible for that, and also good or bad luck. So we'll do our swipe here. The other thing that's of note to us in this community is that fortunately in Northeast Florida, we've had less of an issue than other parts of the state. So if you look at some of the different counties, clearly the three southeast counties in Florida have been hit the hardest, probably because they're centers of tourism and people come from all over the world there, and they're more densely populated than Northeast Florida. But we've been very fortunate, relatively speaking, here in Duval County. Also, there's been a nice drop in the number of cases over the course of the last 6 weeks. So the caseload was probably at its maximum during the last week of December, first week in January. And since then, we've seen drops in new cases that are over 50%. Unfortunately, there's been a plateau of that over the course of the last 2 or 3 weeks. So we're all anxiously looking to see what happens. And the best metric, in my opinion, to understand that is to not look at the total number of cases, because that depends on the number of people who are tested on a day-to-day basis. The best metric is to look at the percent positivity rate. And so you can see here that about a month ago, we were looking at about 8 or 9% in the state of Florida. 2 months ago, we were looking at about 12 to 14% positive in the state of Florida, and we're now down to about 5 or 6%. But unfortunately, I'm not seeing this go down over the past 2 weeks. So we need to see this drop below 5%. It is a little bit better in the 5 counties in Northeast Florida. The 5 counties in Northeast Florida, we're looking at positivity rates 4 to 5% now, which is a little bit better than South Florida. So we're doing a little bit better, but we need to see all these numbers across Florida get well below 5%, ideally down to 1%. At that point, we may start to feel some degree of normalcy. So the other point I'd like to make before we get into our questions is that the fact that we're all vaccinated is unbelievable. It's just an unbelievable technological accomplishment. This is like landing a person on the moon. It really is. And to help you understand that, get the perspective of that, is that you go back to the slide, is that we typically, as a medical community, take between 5 and 10 years to develop a vaccine. And in this case, we've not only developed 3 vaccines that are available to people at this point, not only have we done that, but we've used brand new technologies to do that, and with unprecedented speed and with incredible amounts of information available for researchers. That part is completely unprecedented, the medical response. The virus is not, but the medical response is. And just to give you some perspective, so when H5N1 pandemic flu was first discovered in 1996, we did not have an effective vaccine until 2007. That's over 10 years. SARS actually was a worldwide pandemic that mostly affected Asia, but it was pretty much gone before we had a vaccine. So you don't see any vaccine here. The H1N1 swine flu was pure luck. That became a pandemic influenza virus back in 2009. But we already had something on the shelf for that. So we got lucky. So that way we can respond more quickly. But when you look at these others, whether it's MERS or the avian flu or Zika, basically we were years into this without a vaccine. Ebola, which is absolutely horrible virus, the last major outbreak was identified in 2014, and we just got a vaccine in 2019, despite a huge amount of resources devoted to that. So the fact that we have the ability to be here together, all vaccinated, is really just quite a dramatic, incredible accomplishment of modern science. So let's go to the next slide, and I think that's our first question. Oh. All right, so we're going to get to the question. So as I mentioned, the Socratic method involves asking questions to the audience. And in honor of the Passover holiday coming up this month, I decided that we should have 4 questions. So hopefully that's something that we can all interact on and talk about. So this is the first question I'm going to throw out to the audience. What demographic factors, meaning age, sex, race, et cetera, most strongly influence COVID-19 morbidity and mortality? So there's a lot of discussion about this in the media, as you can imagine. And let me just throw it out. I'm going to pick on Dr. Rothstein to give us his impressions to start.
Among the demographics,
He's our best student in the room. Without putting pressure on you.
okay, I'd say age.
Okay. And what do you say about that? And what about some of the others?
Well, I think that among the others, we know that men are more affected than women. The data that I've seen on race is fairly still ambiguous about whether it's race itself or whether there's other social factors associated with racial differences that are the key player in it. But we know for sure that age is probably the most important. That's why all the vaccines have been limited initially to people over the age of 65 or that have other comorbidities.
So just because the politicians chose to do that, you think they're right?
Well, my feeling about this, Mike, is that the politicians are listening to the scientists
Okay, all right.
and the scientists.
Okay.
So,
So you may be in the minority on that. Al, what do you think?
I think there's other health comorbidities that play factor as well. So people with autoimmune diseases, people that maybe been on chemotherapeutics, people that have diseases that we don't really think that may be autoimmune regulated, like diabetes. And I have a gut feeling that has something to do with endothelial dysfunction, but that's one of my nerd loves.
You sound like a cardiologist now.
So, no, I'm a cardiometabolic specialist.
Okay.
But I think that does play factors as well as age. But I think age does have some factor because that just means the immune system isn't quite as voracious as a young adult is. So I think that does play some of it, but I think the comorbidities play a bigger issue.
Okay. Janet, you want to weigh in?
I vote with age.
Okay.
Just have more.
Dawn?
Well, I would agree that I believe that poverty has a little bit to do with it. as
Poverty?
far as- Yeah,
Mhm.
but I also believe that comorbidities such as obesity, which contributes to the inflammatory process of the endothelium, I believe that has a lot to do with it, adds to that overall process of the illness and not being able to recover from it and becoming sicker.
I think, there may be a viral load issue as well, because if you look at inner cities.
Yeah, and now you're getting into more pathophysiology rather than demographics. But any other demographic things? Do you think any ethnic group is more prone to infection or more likely to die? Do you think that, Sandy shaking her head, what are your thoughts on that?
We were discussing today about, at least in the pediatric population, that the people of color and Hispanics are more likely to have like the MIS-C complication from COVID. So I do think that plays a role. But again, I also think age is probably your top one because, you know, the vaccines were given to 65 plus from the beginning, but also I know, you know, pregnancy, you know, that they're much higher risk for morbidity and mortality than people who are not pregnant. So I mean,
So is there any plausible scientific mechanism for Hispanic patients having worse complications that we know of?
Diabetes, obesity.
Diet,
The access to healthcare.
yeah,
Okay.
poverty.
Well, so it's interesting. So I always like to look at the data, and I'm going to just swipe here because the clicker doesn't seem to work that well. So these are, I actually spend a lot of time looking at websites from around the country. And Florida actually has a really good website in terms of really keeping track of these things on a day-to-day basis. California also has a very nice website that I look at. And the data from California and Florida are actually pretty similar. I look at New York since I'm originally from New York, and their website isn't nearly as good as ours, just to share that with you. But the data are actually pretty consistent across the country. So Dr. Rothstein and others said that age is a big factor, and that's absolutely correct. It is by far the #1 factor. Nothing is even close to age in terms of this. So just to give you some perspective, I think this should show up. Let's see what happens here. So if you're if you're 25 years or younger, your likelihood of dying from COVID-19 is really minuscule. And it's interesting, people have noticed the fact that kids just seem to have very minor illnesses, and that is true. Every virus has its idiosyncrasies, and this particular virus doesn't seem to have a big impact on younger people, which is very different, by the way, than the 1918 flu. The Spanish flu actually was much more lethal in teenagers than it was in the elderly. In fact, there was a study in 1918 that showed that healthy male military recruits that were hospitalized because of the pandemic flu that year had a 7% mortality. 18-year-old young healthy men. And this virus is nothing like that. So in fact, if you look at the numbers, you can see that in the younger group, which we'll say define as less than 25 years old, oops, you have a mortality of only 1 in 4,000. Now, when you get to a little bit older, say between 55 and 70, you have a mortality of roughly 1 in 40. Huge difference. That's 2 orders of magnitude different. And when you get to 85 or higher, when you're in your 80s, your mortality is now 1 in 4. So that's a huge, huge difference. And that is really the most dominant factor in terms of demographics in this disease. But interestingly, even in the elderly, I've had patients who are 80 years old, 90 years old even, that have gotten COVID that have had relatively minor illnesses. And so that's a bit of a mystery as to why some people just experience very, very progressive and severe problems, whereas others seem to be able to fight it off relatively quickly. Probably has something to do with the immune response. I've seen some hypotheses about the way T lymphocytes recognize the virus early on. A lot of the studies that have looked at treatment have shown that people that have high titers of antibodies relatively quickly do well. So there's probably something that we'll learn about it in greater detail about the way the immune system is able to recognize the invader and then be able to elicit antibodies to neutralize it. But that's still being worked out. The other thing that comes up is, about racial differences, and the truth is there's no difference. So if you look at the Florida data, it's identical. So here, this shows you the case mortality rate. So if a white person in the state of Florida gets COVID, the likelihood across all ages of dying is 2.2%. For Black persons, it's 2.0%. But the more important statistic is the mortality per 100,000 population. It's identical for whites and Blacks. So the coronavirus doesn't care what color you are. So that's something you can be very comfortable about. And then as far as ethnicity is concerned, Hispanics actually have a lower mortality rate than non-Hispanics. And probably the reason for that is because they're more likely to get infected at a young age. Again, age is driving that. So age is the key factor. The other key factor, and Dr. Rothstein alluded to that, is actually sex. Sex is way more of a factor than ethnicity. Here you can see that Hispanics have a lower mortality rate than non-Hispanics. And this is, by the way, is not only in Florida, California, the exact same thing. But men have a much worse go at it than women. So have you heard that? Has anybody been feeling sorry for us men? Yeah. So anyhow, that's just an interesting part of the demographics when you actually look at the data. All right. So one of the key elements, as I mentioned to you, is the fact that in Florida, you would expect us maybe to have a higher death rate than most states. The simple reason is that we have more older people. So if you look at the number of people in the state of Florida who are 65 or older, it's 20.5% of the population. The only other state that has a higher percentage of older people is the state of Maine, whereas other very populated states like California and Texas have much younger populations. So we can go back to the numbers, but I think you'll find that we should actually feel pretty good about how we responded in the state of Florida, because our mortality rate is actually lower than a lot of other states, despite having more vulnerable patients. So to me, that's a nice little positive message, and this supports that idea. Again, as I pointed out before, if you look at the total cases per million population, California 90,000, Texas 92,000, Florida 89,000, New York 87,000, Illinois 93,000, Georgia 95,000 per million population. This is per capita. Death rate, California 1336, Texas 15, Florida lower than Texas 1450, New York 2470, Illinois 1800, and Georgia just next door 1646, despite the fact that we have an older population than all those other states. So again, that's a little bit of reassuring news that we're doing something right here. All right, question number 2. Which vaccines have received FDA approval for distribution in the US? Let's see, who should I pick on? I'll pick on Mark.
The earlier ones were Pfizer
Mhm.
and Moderna, and then more recently the J&J Johnson & Johnson vaccine in the last, I think it's been in the last week, has been approved.
Well, any other thoughts about that?
They have EUA.
Ah. So what's the answer? It's a trick question. Sharon, what's the answer?
Have emergency use authorization, but not FDA approval.
All right, so what the numerical answer to that question is?
Zero.
0. There are actually no approved vaccines in the United States. So what's the difference? It's actually a relatively important difference to understand. So typically for the FDA to approve a vaccine, they would want usually a year or 2 worth of data. For COVID-19, they stated that if they have 6 months worth of data, they'll be willing to approve the vaccines. When the product is approved in the United States, it means that the manufacturer can sell it directly to healthcare entities. And if it has a license to write prescription, hospitals, physicians, et cetera. But under emergency authorization, the FDA changed their rules and allowed it to be distributed, these 3 products, and Mark was right about that. There are 3 products that will be available in the United States, but it's only through the government. Okay, emergency authorization means that it can be distributed to the government, and the government then decides how it'll be distributed from there. So the way it actually works, and that was a rule that was put in place just for COVID-19, and the FDA rules were that if you could show us 2 months worth of data showing that the vaccine was effective and safe, then we'll give you emergency authorization, not approval. Pfizer and Moderna are actually putting together their 6-month data, and Dawn and others are helping us collect that. And we believe that Pfizer and Moderna will be submitting that information to the FDA and hopefully getting approval, real approval, in about a month or so. So that'll be something to look out for. At that point, it can be even broader distribution. So then you can go to your doctor's office or any place and get the vaccine. Now, go ahead.
Why did they choose 2 months?
Political, partially. They had such pressure to get things out there as quickly as possible. They did some analyses in terms of whether or not you can find statistically significant information after 2 months. And the experts got together and said, yeah, it's a little bit short period of time, but it's better, it's the best thing we got right now. And because of the severity of the pandemic and the compellingness of having to do something, that's what we'll go with. So to answer your question, it's somewhat arbitrary. There was modeling that was done, but it's always difficult decisions. At the end of the day, somebody has to just make a decision in what they think is the public's interest. But even the 6 months that they're using for approval would be unprecedented. Dawn has done a lot of vaccine studies. Have you ever heard of a study that got approved after 6 months before?
Never,
Right.
never, never.
Right. Now, the other thing to keep in mind is that the number of patients in these studies has never been this large. So, and again, I have to look at that. I don't know the exact answer to this, but if you really to look at safety, you're multiplying the number of patients in the study by the time. So it's really patient years. So we've never had a study, for example, like the Pfizer study, where 44,000 people were enrolled in 7 weeks. It's crazy. It just, the amount of resources that went into this is just amazing. But when you think about it, when you have all those patients, you get a lot of safety information relatively quickly. Of course, you don't know what's going to happen a year from now, but based on what we've learned so far and based on the nature of these products, we don't have any high expectation that there'll be any long-term side effects. And we believe that the long-term side effects of COVID infection are far worse than the long-term side effects of the vaccine. So based on that logic, the FDA chose those numbers.
And a recent study that we just completed enrollment, we did in 41 days, 30,000 patients. That's unbelievable, unheard of.
Yeah, and that'd be the Novavax study.
Yeah.
And we'll talk a little bit more about that. But here in Northeast Florida, we actually had 481,
92, 492.
okay, 492 people participate that here in Northeast Florida at our center, which is just phenomenal, just absolutely phenomenal. So again, we've never seen this number of patients get involved in research so quickly. And for that reason, they've shortened the timelines to really unprecedented levels.
The flu vaccine studies, the H1N1 studies we did historically in the past, 700 patients would be their normal enrollment in a flu study.
The whole study.
Nationwide.
Yeah.
That's how different this is.
Yeah, so some of the questions that would come up about that is when we deal with the variants and you have to make adjustments to the vaccine, do we have to do another 40,000 patient study or not?
Yeah, I don't think so.
Yeah. It just, but the resources that have to go into that are just mind-boggling. And just to do some of the math in terms of the expense. So it was estimated that Pfizer, which completely self-funded their vaccine program and was first to get emergency authorization, probably has already spent $2 billion in the research. I don't feel sorry for them because they're going to be selling this at $20 a shot, and they'll sell at least 1 billion doses. So 20 times 1 billion doses is $20 billion. That's a pretty good return on investment. So don't weep for Pfizer. Okay. All right. So we got this one right. And let's just look at the details. So as I mentioned, the first two vaccines up on this list competed actually very aggressively against one another. I think there's another reason why they got out so quickly is that both Pfizer and Moderna wanted to be the first to market. And we saw here in the research world how they really just continue to crank up the pressure and put more and more effort in recruiting patients and getting things done as quickly as possible. Literally, when they were collecting the information, they were texting me and emailing me like several times a day to make sure that I would sign off on the system so that they can transfer the data to the FDA. It was just absolutely crazy. But it turns out that Pfizer beat Moderna by 1 week. And they both use a similar messenger RNA platform, which is just a super cool way of developing a vaccine, in my opinion. And this has never been used before. This is a brand new way of making vaccines. And basically, for those that are not familiar or haven't read about it, what the messenger RNA platform does is put the coding for the spike proteins or portion of the spike proteins from COVID-19 into a little sequence of messenger RNA. And they're able to do this because as soon as the virus was identified and the genetics were shared, you can track all the different genes and then find out how the messenger RNA would look for parts of the spike protein. So they took unique parts of the spike protein that would attract antibodies and that would allow the antibodies to prevent the spike protein from infecting human cells. Just to remind everybody, coronaviruses are ubiquitous. There's always been coronaviruses. There are only 7 coronaviruses that have been proven to infect human cells. The vast majority of coronaviruses cannot infect human cells. They infect human cells through the ACE2 receptor, and 4 of the 7 coronaviruses that can affect human cells cause very benign symptoms, very low-level cold and flu-type symptoms. But the 3 that cause more severe symptoms are SARS, MERS, and SARS-2, which is COVID-19. So it's the exception to the rule that a coronavirus can cause severe illness. So with that knowledge, the people that made the messenger RNA platform came up with the concept that if we can prevent the spike protein from infecting a cell through the immune system, that should be an effective vaccine. But again, the phase 1 studies that were done really, really quickly showed the ability to create antibodies, but they were relatively small amount of patients, and they didn't have any of these patients exposed to the real world. So it was a real big unknown going into the studies exactly what would happen. And the actual results were just astounding. 95% efficacy rates, and I'll show you the data, but it's just absolutely astounding in how quickly everything happened. So the enrollment for both of these studies, they started within a week of each other, the studies. I think that if my memory serves me correctly, we started the study on July 27th, and enrollment stopped in the Pfizer study for most people by September 10th. And then we had to get everybody that came in swabbed if we were concerned about the possibility of COVID infections. And then by the first week in November, they announced that we were going to get all the data in and submit to the FDA. And they submitted to the FDA in the middle of November, and they got approval on December 17th. The FDA then told the CDC that we have approval, and now you tell the states how to get the doses. And literally within hours, 2 million doses were shipped. So it's really just amazing. The other thing about the messenger RNA platform is that because of the intrinsic instability of the messenger RNA, which is a good thing, I'll explain that in a second, you have to store this either frozen or deep frozen. So there's been some concerns. I don't think this group of healthcare providers particularly has those concerns, but people say, oh my God, it's genetic material, it's going to change my genes and all this. So just to reassure people is that messenger RNA doesn't even get into the nucleus of cells, it can't. The other thing is that messenger RNA is the way our bodies normally work. It's like our body's email. So our cells are constantly sending out little messages, and the cells are responding to messages. And because they're constant messages and the body doesn't want the cells to get confused about the messages, there are enzymes that are constantly breaking down messenger RNA. So the bigger challenge for these vaccines was to put them in a protective coating, a lipid nanoparticle that allows them to get to a ribosome and actually create pieces of the spike protein, which elicit an immune response. So this is just absolutely astoundingly cool technology. And both the Pfizer and Moderna vaccines basically use it. There's some subtle differences that I won't get into unless there's questions about it. But the bottom line is that both of them had 95% efficacy. And then most recently, as mentioned by Dr. Emis, is the Johnson & Johnson vaccine was given emergency authorization. And that uses a technology that's been around that other vaccines have used, which is it's a adenovirus that cannot replicate itself, that has been given the ability to produce COVID-19 proteins. So basically the genetics of the virus were altered in a way so the virus can get into our body and then produce the spike protein that our immune system then recognizes as foreign and protects us against. And I'll show you more about that. But this is a technology that has been used for other vaccines. And it's also more akin to the technology that's been used by AstraZeneca for their product that's been approved, and some of the other products that we know a little bit less about from China and Russia. So speaking of which, so the Oxford AstraZeneca vaccine did get European approval a little bit after the messenger RNA vaccines. But as you can see here, it's unlikely to receive FDA authorization anytime soon. Most of the work that was done for AstraZeneca was done outside of the US. There were a few sites in the US, and they actually stopped their studies in the US due to a side effect. And I don't think they'll have enough US data to get FDA authorization for that anytime soon, maybe at some point. But at this point, I wouldn't expect that AstraZeneca will be available in the US. Although it is available in the Caribbean now, it's available, I think I've read 30 countries already. So there are places where you can get it. Then you've heard about perhaps the Sputnik 5 vaccine, which was tested on President Putin's daughter. And we don't know a whole lot about it other than the total amount of study patients that were exposed to it before it was approved by the Russian authorities was about 250. Okay, so just think about this. Here at this center where we're sitting right now, we've dosed about 1,300 people in vaccine studies. So in this room, we've dosed more people than the whole Russian program. So it's pretty amazing when you think about the level of information that goes into these things. But the Russian vaccine might have been testing more people at this point, but Putin assured us that it was safe. His daughter took it, and she only had minor fever, chills, and vomiting, and she's still alive. So they distributed, and they claim 91.4% efficacy, although this has not been subject to peer review. And then there are 3 products that the Chinese are licensing to people that we just don't know nearly as much about. And I'm sure at some point they'll get to a peer review, but it's just a whole different system in terms of the level of scrutiny and the amount of testing that's been done. Keep in mind that this is just a small percentage of all the vaccines that have been proposed. There's actually 145 vaccines that have been registered with the WHO. So a lot of companies have tried to come up with concepts. And to my knowledge, last time I read about of the 145, about 25 have been tested in humans at some level. So again, 3 authorized in the US, probably a couple more to come, 145 around the world, and then some of these others that will probably be the vaccines that are used outside of the US in large numbers. The other one that Dawn alluded to is called the Novavax program, and that actually uses a really cool technology as well. So instead of using messenger RNA to create the spike protein, what this technology does is actually makes the spike protein using insect virus. And it basically is genetically engineered spike protein that's purified and put in lipid nanoparticles. And then it's combined with something called an adjuvant that allows the very small amounts of spike protein to elicit a major immune response. So using this concept, and the adjuvant is called saponin, which is a way that the vaccine presents the protein to the T cells in the immune system. So the T cells are the cells that recognize the antigen, and they tell the B cells to produce antibody. And because of this combination between an adjuvant and the protein, you're able to use lower amounts of protein. So one of the theories is that you may have far less side effects with this because the way the immune response is elicited is fundamentally different and doesn't require as much protein. So we'll see. But the interesting thing about the Novavax study, it's actually a US-based manufacturer out of the DC, Maryland, it's based in. But they did their initial studies in the UK and South Africa, and they actually reported their data, which is over 16,000 patients, that showed that it works. But they also showed that it doesn't work as well for the South African variant as the wild type variant or the UK variant, which raises some interesting and somewhat concerning elements. But the fact is that this is another product that we've used here in Jacksonville. As mentioned, we had 490 patients involved in this. So far, so good. We're not seeing any major side effects. People are doing well. The study that we ran has a 2 out of 3 randomization to active vaccine right away. And then following the initial randomization, 2 to 3 months down the road, people can cross over. So anybody who did not get the vaccine initially will cross over to the treatment group after about 2 or 3 months. So we're doing it right now here in Jacksonville and one of the major centers around the world that's participating. So kudos to the team. And I think I mentioned this. The couple of interesting things is that the overall efficacy of the Novavax in the study that was done overseas was 89%, but South African variant was more difficult to treat. And we'll talk a little bit more about that. So that is one level of concern for the future. All right, question number 3. How do the FDA and others calculate the efficacy of viruses? Anybody want to volunteer for that? Like, where do these numbers come from?
Data.
Okay, well, we'll go with that, absolutely. Mitch, want to get more specific?
Not really.
Smart man.
Is it based on for instance, in the Pfizer study, it's the number of patients that they actually enrolled, and then since it was a 50-50 randomization, they're looking at the number of patients on placebo that actually got the COVID. And they're comparing that to those that received active drugs that got COVID.
You're very well trained. Everybody tell you that?
I am. Somebody trained me well.
That's exactly right. So, you know, when you hear all these numbers, like, what does it mean to you? So the first thing I'll tell you is that it's astounding. Like, Dawn, what's the usual efficacy rate for a flu vaccine?
Oh, it's probably 70%.
I said that? So I heard from this side of the room. You said that.
I did not say that. Sorry.
That was right. Whatever you said was right.
That's not too low, so I think.
Okay.
It's 60 to 70%.
Dr. Lopez knows how to throw his voice in yet.
I wish I said that.
I'm sorry. I
60
cut you off.
to 70%,
Yeah,
somewhere
exactly.
in that area, yeah.
And we're usually happy about that.
Oh my God, thrilled.
Right, right. So what was your reaction when you heard 95% efficacy?
Couldn't believe it.
Yeah.
You just don't see that. So the shingles vaccine is very much like that, Shingrix,
Yeah.
with a high efficacy rate.
Yeah,
That was amazing. That was the first amazing one we ever saw.
it's just mind-boggling. Yeah, I was completely blown away when I heard that. So let me show you how they actually calculate it.
Okay.
So every time, this is actually interesting. I find this interesting. Is when you're working with the FDA, the way the process plays out is that the FDA gets panelists from around the country who are experts in whatever the area is that's being evaluated. And they'll, in the old days, they would sit in the same room and hash out the discussions and the data, look at it. Obviously, it's done virtually these days. But what the FDA does is provide what they call a briefing document, which has everything you need to know about the particular product in one document. And so, and I think this is unprecedented, at least in terms of public access, is 2 days before the advisory meetings for all 3 vaccines that have been emergency authorized, they release that publicly. So you can actually get 48 hours, and I was like thrilled to be able to read these things, to look at it as the advisors were looking at it. So you kind of look at it in real time. And so, as a researcher, of course, I read through these things, and you find a lot of little tidbits. Like, I can tell you how many people in the Pfizer program got pregnant while they were in the study, 25. So there's all these like crazy little tidbits and trivial things and whatnot. But that's how these numbers come. So to Dawn's point, she's exactly right. So what you're looking at when you come up with the efficacy rate is you look at the number of cases of COVID you had on placebo. And if the vaccine didn't work at all, it should be the same number in the treated group, right? If the vaccine was 50% effective, you should see half as many cases as you saw in the placebo group. If the vaccine is 70% effective, you should see 30% of the patients in the treated group versus the placebo group. If it's 90% effective, then you would only see 10%. But here for the Pfizer study, you can see that in the treated group, there are only 9 cases. And in the untreated group, there were 169 cases. Just a massive difference. And that doesn't even speak to the severity. It just means that you had a positive PCR. And so that, when you calculate it out, is 94.6%, or the famous 95% that you're hearing.
They take into account like when the infection was
Yeah, good point.
could it be like, really quickly, and they may have had it previously.
So we'll get to the previous thing in a second, which is a really interesting point. But the way these things work is you define the primary endpoint before you even start the study. And the primary endpoint for Pfizer and Moderna was looking at infections about 2 weeks after the second injection. So the actual efficacy was based on the number of cases that occurred 2 weeks after the second injection. For the Johnson & Johnson study, since there's only one injection for that, they had what's called co-primary endpoints, which is looking at infections at 14 days after the injection and 28 days after the injection. I'll show you those data in a second. That's a really good point. So going back to this, they broke it down by older and younger people, and you can see basically the same efficacy here for the Pfizer vaccine, whether or not you're older or younger. So really just tremendous information. Question? Okay. So the other thing that I find just absolutely amazing is this analysis. This is called the Kaplan-Meier curve. It's a very, very famous way that we look at the progression of any particular medical intervention and its effects. And in a Kaplan-Meier curve, you compare the treatment group with a control group. So in this case, the control group are unvaccinated people who are depicted in red, and the treatment group are people who are vaccinated that are depicted in blue. So 0 time is when they got their first injection. And this is looking at the number of cases. Each one of these boxes is a case of COVID. So in the blue, it's a blue circle. In the red, it's a red square. And you can see that for the first 7 days, both the injected people and the placebo patients are getting cases. And you can see how this is going up. But somewhere between 7 and 10 days, all of a sudden the injected people stop having cases, whereas the people in the placebo arm are having cases in a linear fashion based on their exposure. Do you see how dramatically those curves separate from each other? Mitch, when's the last time you saw a medical intervention that has that kind of separation?
Never.
Mark, have you ever seen that?
I haven't seen it.
Al?
Never.
Yeah, it just doesn't happen. So that's so satisfying. And when you look at data all the time, like I do, it's just mind-boggling that you see that. And so the take-home message is that there is really good protection from the messenger RNA products about 10 days after your first injection. Now, the question has come up about the second injection. How necessary is that? Is the timing important, et cetera? So I like to think of a second injection as a booster. because we know that you get really good protection from the first injection, but we don't know how long it's going to last. So the second injection could be thought of as a booster over time. So really, really cool.
So I have a question.
Yes.
A friend of mine just texted me telling me that she's a New York nurse. She got COVID. She just got.
That's the first problem.
But a friend of hers got their second injection a month before and now has COVID. So for those people who are in the 5% category, I mean, I know the CDC is saying don't go check your antibodies, right? I think that was one of the last things I heard. You don't need to run out and check to see if you have it.
Right.
But if you happen to go run out, see that you donate blood and you don't have antibodies, is there a third booster that should come into play?
Yeah, so there's a lot of nuance to that question. So first, I'd be curious to know the details of that person's infection in terms of symptoms. Were they sick before? Some people never get rid of the virus, or it takes a long time for them to get rid of it. So there's always a possibility that person had the virus before they got their first vaccine. So that's one issue. As far as antibodies are concerned, there are lots of different antibodies.
Right.
So no antibody test is exactly the same. And the antibodies that you get from being infected are different than the antibodies you get from getting the vaccine. And depending on the type of testing you're doing, they may not correlate. So we have a lot of patients that get antibody tests, and it's a little bit of a crapshoot if it's meaningful. So it's really hard to guide people based on that. Bad luck though. But the good news is that even people that develop COVID based on PCR positivity that have been vaccinated tend to have very mild illnesses. So hopefully that's the case for your friends. All right, so moving on to the Moderna, you can see that we saw something very, very similar. As I mentioned, 95% for Pfizer, and Moderna was really pretty similar. Their study wasn't quite as large. The Pfizer study, as I mentioned, had 44,000 people. The Moderna study had about 30,000, but the results are similar. People vaccinated had 5 cases over the course of 2 months of observation median time versus 90. Again, if that number was 9, that would be 90% effective. If it was 45, it'd be 50% effective. In fact, it was 95% effective. So very, very similar results. And again, we see excellent results in both younger and older populations. They had no cases of vaccinated patients over 65 that were positive amongst 3,500 people that were studied. So that's really, really impressive. And then if you look at the Kaplan-Meier curve, you see the really identical results. You see that there's slightly different graphing system that they use between the manufacturers, but starting at 10 days, these curves separate, and dramatic difference. So again, really, really impressive. All right, and then this is the Johnson & Johnson information. So the Johnson & Johnson information is a little bit different fundamentally in that it's a one vaccine product, one shot. It was done at a different time. Remember, this was delayed about 3 months or so after the start of the Moderna and the Pfizer program. There's pluses and minuses to that. When the Johnson & Johnson folks were running their study, it was actually through their Janssen division. When they were running the study, it was already a huge problem in the places where they were running it. Whereas when Pfizer and Moderna were running it, it wasn't quite as widespread. COVID infections weren't quite as widespread. So Johnson & Johnson is, again, a massive study with over 40,000 people, but they had way more cases overall. The other thing that's interesting about the Johnson & Johnson program is that it was done in South Africa, where we have a variant to deal with. It was done in Latin America and the US. So about, if I remember correctly, 44% of the cases were from the US, about 40-some-odd% from Latin America, about 20%, a little bit less from South Africa. But when you look at the numbers, you can see that the placebo group had 348 cases. So if this number was 35, that would be 90% effective, but in fact, it's 116. And that comes out to be 67% effective. Now, they had what I mentioned as co-primary endpoints. So when they submitted this study to the FDA initially, they said, we're going to look at infections at least 14 days out and at least 28 days out. But you can see the numbers are pretty much identical between those 2 endpoints. So there was not a whole lot of difference between 14 and 28 days. Again, the breakdown by age was no different, but those numbers are a little bit different. So some people have been a little concerned about that, suggesting that maybe the Pfizer and the Moderna products are simply better. But the interesting part about the Johnson & Johnson product is that they too had a very impressive Kaplan-Meier curve. So maybe this slope is a little bit higher in the placebo arm than it was in the others, but you can see that at 14 days, maybe a little bit later, those curves separate really, really nicely. So there's no question that this vaccine is effective. So that's really encouraging. And if you look at this particular slide, there's something else that comes out that's really, really interesting, which is that 10% of the people in this study had COVID before they even started the study. At least 10%, because to your point, not everybody's going to be antibody positive, but this looked at the number of people who are antibody positive. So the question comes up then, does the vaccine do anything in antibody positive people? And two, do antibody positive people get COVID? Two good questions, right? So we can actually answer that question now based on this really nice database. So this looks at the number of patients in the placebo arm that did not have antibodies that got COVID during the course of the follow-up, which again was a little over 2 months. It was 513 out of 21,000. That's over 2%. It's about 2.5% of people in the placebo arm got COVID within the next 2 months after being injected with placebo. As mentioned, the rate of getting infected if you were actively vaccinated went down quite a bit, but it was about 0.8%, a little bit less than 1%. But now you go down to the placebo group of people who had antibodies, and it's only 4 patients out of 2,000. Okay, so if that was 20 patients out of 2,000, that'd be 1%. So this is about 0.25%. So compare the 0.25% with over 2.5%, and you can see the fact that previous infection really protects you by an order of magnitude. It's not impossible to get reinfected, but it's really, really unlikely. So when you think about it that way, taking the vaccine, if you've been infected, should, in my opinion, be looked at more as a booster rather than a primary means of treating it. Because again, that percentage was similar to what you saw for people who are vaccinated.
Is that number 4? Is that they had the antibody and they got the vaccine, or they didn't get the vaccine?
Those are people who did not get the vaccine. Or out of 2,000 who did not get the vaccine, but who had COVID before got infected.
So in the J&J study, did they bring them in, screen them, and vaccinate them? They tested them and vaccinated them on the same day.
Right.
And so they wouldn't have known if they were COVID positive or not at that moment.
Exactly.
Right?
All the studies like that. The studies that we did.
Ours is the same way.
Right. If you, so for example, in the Pfizer and Moderna studies, which we participated in, patients were asked, do you have COVID? If they said yeah, they weren't allowed to be in the study. If they said no, we put them in. 2% of all those patients had COVID in Pfizer and Moderna, and 10% at least in Johnson & Johnson had COVID already.
Which brings up an interesting point. So if they had COVID, that's kind of their initial event, and then they got the J&J shot, that was their booster, is what you're saying essentially.
Exactly.
But then in Pfizer and in Moderna, if they had COVID before, they were still allowed to get their first shot and their second shot.
Right. Right.
So they were really boosted.
That's how we learned. And that's why some of them had actually very severe side effects, because
Exactly.
their immune system was really highly revved up. All right, so we're going to move on a little bit quickly now, but I find this is really interesting. So Johnson & Johnson doesn't have the same efficacy rate, but it's really good at protecting against severe problems. So this is now looking at more severe issues like dying. And you can see that the Johnson & Johnson vaccine actually protects people from dying. In fact, there were no deaths due to COVID-19 for people who are vaccinated versus 7 deaths in the placebo group. And if you look at all-cause mortality, you can see that there was a reduction in all-cause mortality if you took the Johnson & Johnson vaccine. So that's reassuring. And this just shows you the difference between the different parts of the world. Mentioned the US efficacy rate was 72%. In South Africa, where they were dealing with that variant, it was 64%. And Brazil, where they have some variants, it was in between about 70%. All right, so now the final question, or the 4 questions. How do we get to herd immunity in the US and Florida? And when do we get there? So obviously that's what we're all shooting for. And once we have herd immunity, we believe the virus should go away pretty quickly. probably within weeks, because there'll be no place for the virus to go. Viruses need to have a host. They can't live on inanimate objects for very long. So they need to infect something or somebody. So where are they going to go once we get to herd immunity? So that's going to be the question. So actually, let me open up to the audience. I'll pick on you again, Mitch. What's your prediction and why?
So so herd immunity, first we have to figure out what percentage of the population has to be exposed or vaccinated to establish herd immunity. And I think most people put it somewhere between 60 and 80%, so about 70%. So if we have 20 million people in Florida,
21 million.
yeah, so about 14 million need to be vaccinated. 14 people would need to have either been infected or
There
vaccinated.
you go. Exactly.
So,
I like your math.
well, that's about where it ends. The rest is guessing. So I say probably 6 months.
6 months. Okay. Sandy.
I agree with him. I
Very
think.
smart.
No, I think that I read something that CDC was saying they want 70% vaccination. But again, I mean, what percentage are children of the US population? Around 20%, 25%. So we don't know anything on the study. I don't know anything on the studies for them yet. You deduct them, again, Dr. Sapolsky, who I believe is in the group in virtual land. So there's a good, probably 10% hardcore who refuse to get the vaccination. So now you're up to 32%. So we just kiboshed our 70%. Because then you also, like she said earlier, that you have that group that's wishy-washy and doesn't know if they're going to get the vaccine. They're going to let us be the guinea pigs first. So it's hard.
Yeah.
So was the question, when do we think we'll be vaccinated? Or I mean,
When
sorry,
do we get
herd
to herd
immunity.
immunity? Yeah.
Spanish flu took 4 years. My guess when this all first came out, I said this is going on for at least 2 years with all of our rapid technology.
All right. Al, you want to prognosticate for us?
Yeah, I think she's pretty correct. I think it's going to be the end of 21. I think it's not going to be until late winter when we'll see enough people either infected or vaccinated for us to really see it.
Okay.
The hope is the summer, but I think that's just too hopeful. I don't think we'll get enough vaccines out.
Don?
I agree with him.
Okay.
Will it burn itself out ever? Like,
Yeah, well, that's an interesting question. So that gets to when you say burning itself out, it means that it varies in a way that it becomes so benign that you don't even notice it. A lot of viruses do that. When they mutate, they can mutate to more benign forms that are rapidly passed and people get to herd immunity quickly. I actually think we'll hit herd immunity by the end of April.
No kidding.
And anyhow, I'll show you why. So anyhow, this gets to Mitch's, he was right spot on in terms of his calculations. So remember, there's 2 ways you can get immune, either get vaccinated or get the virus. A lot of people have gotten the virus. So that's the unknown, is how many people have gotten the virus. We know how many people have been diagnosed with infection, but we really don't know how many people have gotten the virus and how many people are immune. And so that's the question. That's why it's really hard to speculate how quickly that's going to occur. Keep in mind that, as I mentioned, coronaviruses are ubiquitous. So for a lot of us, we've been exposed to coronaviruses. We may have some intrinsic immunity. We'll talk about that in a second. But anyhow, this is just showing some of the variability in the day-to-day reporting of the Florida website. So I don't so much look at the day-to-day, but if you look at the total cases, we've documented close to 2 million cases in the state of Florida. So that, as I mentioned, probably about 10% of the population. So how many cases have not been officially documented? So in kids, probably a lot of cases have not been officially documented. I'll tell you an anecdote. My college roommate who lives in Toronto called me in a panic 3 weeks ago that his son with 3 young kids came down to Disney from Toronto, and against my roommate's advice, and they came down here and they had to get tested before they could go back to Canada. And the 5-year-old, he has 1 3 and 5-year-old, tested positive, completely asymptomatic when they try to get back to Canada. So they wouldn't, of course, they wouldn't let the kid on the plane. And my roommate's in a tizzy, said, "Can he stay with you?" So I have a beach house. I said, "That's fine. Let them stay at the beach house. They can quarantine there." So they all go to the beach house. I said, "First, before you go to the beach house, though, test again. Maybe it was a fluke." They test again the next day. The 5-year-old is positive. Everybody else in the family is negative. Everybody is asymptomatic. So they go to the beach house, and they stay there for about a week. Over the course of the week, all 3 kids are running around, feeling great, just doing their thing. Both parents start to get cold symptoms. Now they're in their 20s and they had minor illnesses. And then my college roommate's son figured out that, well, there's no way he's going to be able to get on a plane, so he rents a car and drives to Canada, and they don't check you when you drive across the border, only when you fly into. So so much for all these crazy restrictions. So yeah, my point is that those are 5 cases of coronavirus. Only 1 will be documented.
Right, my neighbor's same way, a family of 5, only 1 tested, but we know at least 2 others have it.
Right. And for the kids, none of them you wouldn't even know it. The only reason they were tested in the first place is because they had to be to get on the plane. So that's the unknown. We just don't know that. So it's all guesswork. But if you go back to the numbers here, if we have 2 million documented cases, I think it's fair to say that we have at least 2 million undocumented cases. And I personally think that's closer to 4 million undocumented cases. That's pure speculation on my part. So you start putting those numbers together, and you see that we're maybe a lot closer than you think. So as I mentioned, 2 million infected, at least 2 million infected, but not tested. And then we have 3 million vaccinated now. So you add that up, and so we have 7 out of 21 million Floridians who are immune right now. So that's at least a third. It's probably more than that. We're probably around 40% right now. And again, depends on how you treat kids. If you think of them as just so unlikely to get really sick from this virus for whatever reasons, then we could be close to 50%. Yes, Mark.
What about the natural infections losing immunity?
Yeah.
They're not sure about that.
Yeah, so that's interesting. Well, there's 2 parts of that story. So again, my guess is that 35 to 40% Floridians are already immune. As mentioned, we need probably about 70 to 80% to get to herd immunity. And we're going to see a lot more people vaccinated because of now Johnson & Johnson coming out with theirs. Merck is helping Johnson & Johnson manufacture. Pfizer just got somebody to help them manufacture. Moderna. So there's going to be just incredible manufacturing surge, in my opinion, over the next 6 weeks. And hopefully it's going to open up to virtually anybody that wants it. Obviously, there'll be people that don't want it, but they're going to get sick. They're going to get sick, and then they'll become immune. Hopefully, they won't get too sick. And then this question about natural immunity, and I've heard that thrown around quite a bit. I was listening to an epidemiologist from Johns Hopkins actually this week talk about that. What that really means is that previous exposure to other coronaviruses will protect you against COVID-19. So your immune system has enough memory to say, okay, well, this spike protein is not exactly what I saw before, but it's close enough. I'm going to send out antibodies and protect you. So that's a concept. Versus people that lose the ability to fight it off. And I don't think that's going to happen in very many people in a short period of time. In fact, I read a study showing that people in their 90s who were exposed to the pandemic flu in 1918 or in the early 1920s still have memory for that particular virus. So there's probably some T cell or 2 that's still hanging in there that knows that. And that gives you a big head start.
So Mike, when we reach herd immunity, since 25% of the antibody positive patients won't be tested, we'll recognize that by a fall in the new case positive rate?
Yes, I think so. So the real concern, my concern is how the variants play out. So I'm actually worried that we haven't seen that positivity rate drop in Florida and other places in the last 2 weeks. And that may be because we're starting to see some variant cases. In South Florida in particular, the UK variant is prevalent. The South American variants are been seen in South Florida. The California variants have been seen in South Florida. The New York variants have been seen in South Florida. Fortunately, not the South African variant. There's very, very few and far between. And that's the most concerning because that has the greatest genetic difference from the wild type COVID-19. So that, if you look at the information I showed you, that's probably least likely. The South American virus is least likely to respond to the vaccine. And the most concerning, but the good news is that we haven't really seen it in the US for no reasons.
What about spring break? Duval County starts their spring break this week, are they anticipating a surge like they did after winter break?
It depends on people if they behave themselves. And we know how the virus spreads.
We have the college factors removed from that because a lot of the colleges are not doing spring breaks. They're going straight through. So what we have is middle school and high school and elementary and college students, which seem to have affected adults more, is out of the picture theoretically.
But it will be a challenge, no doubt.
So you're saying the fact that the case rate isn't decreasing in the last several weeks is because the vaccine's not protecting you?
No, the
against
last,
variants?
it's like the last 2 or 2.5 weeks. In other words, we saw a decline. In Florida, we were like at 12, up to 15% in late December and in early January, and it came down to like, 5 or 6%. But it's hanging out at that 5 or 6%, not going down further.
Is that you're saying it's a vaccine not being protected?
No, the cases aren't occurring in the most vulnerable people. So the death rates are coming down still, but it has to do with the incidence and whether or not that'll eventually cause some problems and translate to more hospitalizations. Hospitalizations have come way down. Hospitals are quiet again. The easiest way of finding out how active this is just do rounds. So I still do rounds. And in December, the hospitals were 30 to 50% full of COVID patients. And now what you said, Al, was 10% COVID patients now?
Yeah, we're down from almost 100 to 150 per hospital. We're down to a total of 150 in the city.
Yeah, the whole city, amongst all the hospitals. So huge drop from where it was 3 months ago. So, well, yeah, again, I'm hopeful. I'm hoping also, quite frankly, points that were made is that a lot more young people have been exposed already than we know because they tend to have mild illnesses. We're not really testing them. So we'll see. But it is a point of concern. And I'm anticipating that probably everybody over 40 will have access to the vaccine in Florida within the next 3 weeks, based on projections of manufacturing capabilities.
Have you received any information about how long the antibodies are staying in the body after Pfizer and Moderna? And are they going up? Are the levels going up, or are they staying steady?
The levels go down over time if you're not further exposed, but they haven't gone to zero. And so the real question is how quickly you can elicit immune response in 6 months or a year. We don't know the answer to that. But if you have T-cell memory, you should be able to mount in a response really, really quickly.
There's a question on your thoughts about wearing masks when outside.
Yeah. So wearing masks it's an interesting discussion point. Unfortunately, it's become more political than scientific. So just to give everybody the quick scoop on masks. If you really want to prevent the virus from being transmitted, you need to wear an N95 mask. If you wear a paper mask or a cloth mask, the pores of the mask are as big as the virus or bigger than the virus. So you'll have an imperfect way of preventing that. Having said that, a paper mask or a cloth mask is better at preventing somebody with the infection from spreading it widely, because some of the virions will get caught up in the fabric of the mask. But if you really want to do it right, you need N95. But if you're outdoors and you social distance, the likelihood of you transmitting the infection is really, really, really low. So I kind of laugh. I'll go to a restaurant and I'll see people hanging out at the bar wearing masks and then hugging each other. And then I'll see people outside waiting for a bus on a park bench by themselves wearing a mask. Or I'll see the football players that have to wear a mask, and then when the player comes from the sideline, the coach takes down his mask to yell at the player in his face. So, but when the coach by themselves, they're wearing a mask, but when they're yelling at the player, they take down the mask. So it's clearly a fundamental misunderstanding how masks are supposed to work. But again, what we're doing here, to me, makes a lot of scientific sense, which is we're staying apart. So again, no kissing, no sharing drinks, and we'll be okay. Plus, we're vaccinated. Does that answer the question?
Yes, thank you.
Any other questions? You're our co-chair.
Are we over? Are we done?
Well, I'll give you the last word. How's that?
Well, I learned a lot. I mean, I probably have another million questions for you, so I'll pretend to have chest pain and come in and book my 30-minute appointment with you. But that was great. That was interesting. very helpful. I hope everybody on virtual land, thank you guys for joining us tonight. We do have a part 2 of our program next week with Dr. Wendy Sapolsky and Dr. Jennifer Fulton. They will be discussing the impacts of what's going on with the virus for children and adults and some of the most common questions that people are wanting to know. So on behalf of the Jacksonville Federation and Foundation, I thank you for joining us. And it's a wonderful charity that helps out for the Northeast Florida and donates to for Northeast Florida, Israel, and across the world. And if you have not already donated to their 2021 campaign, we invite you to participate in that. And again, thank you. That was wonderful.
Thank you. Thank you. Just a quick plug for my organization. We are running trials now in all different areas. We are actively enrolling in 150 trials. So for healthcare providers out there that want to get involved in research, come by, let us know. We're actually about to start a chikungunya vaccine study, which is another virus we can talk about another time, which is very common in Latin America and hopefully will not become common in the United States, but we will be better prepared for that because we're actually studying the virus now. So just an example of some of the research that we're doing here. And of course, I every once in a while get back to my real job as a cardiologist, and we're doing a lot of studies on cholesterol, including novel lipid molecules. Dr. Emis has worked with us on some neurological things. We're actually doing Alzheimer's research. Dr. Rothstein is a critical care physician who's working with us on a phase 1 unit, where we actually take molecules that have been maybe only used in a few people and watch how well people do when they have these novel therapeutics that we actually do here in Jacksonville, Florida. So we're doing a lot of cool things here, and it's really open to the medical community. So we're more than happy to get people involved. So thank you for being part of this, and this is fun. And be careful.
Thank you so much, Dr. Korn. I had a couple of requests for the websites that you shared earlier in the presentation, and I just want to let those of you know that reached out. I will follow up and get those to you as soon as I can. So thanks for being with us tonight.
All right. Well, thank you.
Thank you. Night, everybody.
Hi.
Thank you.